Hi, my name is Jennifer Mason. I'm an assistant professor at Clemson University. Today it is my pleasure to tell you about research in my lab looking at the repair of DNA during replication. Prior to division, our cell must first copy all of its DNA through a process called replication. During this process, the strands of the DNA molecule separate, are copied, ultimately resulting in two identical DNA molecules that are then passed on to daughter cells during division. However, factors in our environment, such as UV light from the sun or ionizing radiation such as X-ray, both damage and can even break DNA, stopping replication. If cells are unable to fix the DNA damage, this can have severe consequences. DNA contains specific instructions. DNA damage that changes even a single letter can have a dramatic effect on the underlying message. Furthermore, if unrepaired, DNA damage can result in cell death, and if misrepaired, cells can accumulate mutation, resulting in human disease such as cancer. Thankfully, our cells have DNA repair proteins that both protect and repair DNA during replication, and my lab is interested in understanding how these DNA repair proteins work by studying three different mechanisms. First, we want to know how DNA repair proteins localize and protect replicating DNA, how they repair the underlying DNA damage, and then finally, how they promote replication restart once the damage has been removed. And so to do this, we use human cell culture models and an experiment called the DNA fiber assay. During this experiment, we add modified DNA letters called nucleotide analogs to growing cells, and these are immediately incorporated into replicating DNA. We then can take the cells, put them on slides, and break them open to release the DNA. We then tilt the slides to allow gravity to spread the DNA across the slide, and use a process called antibody staining to add fluorescent molecules to our modified DNA, and then this allows us to visualize them using a fluorescent microscope. By using this experiment, we can measure the impact of both different DNA damaging agents as well as DNA repair defects on replication. By using this experiment with a variety of other different types of experiments, we hope to increase our understanding of how cells protect the integrity of DNA during replication, understand how defects in these pathways result in human disease such as cancer, with the hope that someday we'll be able to develop therapeutics that specifically kill cancer cells with high levels of replication stress. Thank you for supporting EMGS and listening to me tell you a little bit about my research.